Imagine consuming a large, succulent steak. As you are eating this steak, enzymes inside your body are working to break it down into usable forms of nutrients. Enzymes are not only present to help break down food, but are involved in almost every cellular process. They are catalytic molecules, meaning they increase reaction rates. Most enzymes are proteins consisting of amino acid chains, but some are RNA molecules called ribozymes. In this episode, we will explore the structure and function of enzymes. These cellular machines decrease the activation energy, or the energy required to start a reaction. To lower the activation energy, enzymes attract a substrate, or reactant, and position it so the reaction is initiated. Without these catalysts, reactions would proceed much slower. Enzymes attract substrates to specific locations on the enzyme where the reactions take place. These locations are grooves called active sites. First, a substrate binds and fits into the active site. An enzyme-substrate complex is then formed, where the enzyme changes shape to hold the substrate in place. Now, the enzyme is ready to convert the substrate into products. Let's use a gumball machine analogy where the gumball represents the product. The machine is the enzyme, while a quarter is the substrate allowing the product to be retrieved. Only a quarter will fit in the slot, not a penny or a dime. Likewise, the substrate has to be specific in order to fit into the enzyme's active site. The gumball machine can continue to accept quarters and produce gumballs, just like an enzyme can be used for multiple reactions. Environmental conditions, such as temperature and pH, must be within a specific range for these reactions to proceed. The reaction rate is the fastest in this range. For example, if the pH in your stomach is outside the range of 1 to 3, the digestive enzymes will denature. Not only do enzymes require optimal conditions, but some depend on a helper molecule called a cofactor. A cofactor that consists of mainly carbon atoms is called a coenzyme. These molecules allow the enzyme to function. Even if helper molecules are present, some reactions may not be able to proceed if an inhibitor is also present. An inhibitor is a molecule that affects the enzyme by slowing down or stopping a reaction. One type of inhibitor is competitive and blocks the substrate from binding because the inhibitor binds to the active site. This would be similar to a penny being stuck in the slot of the gumball machine, preventing the quarter from fitting properly. The second type, a non-competitive inhibitor, binds to a location that is not the active site. This binding causes the shape of the active site to change, but still allows the substrate to bind. Allosteric regulation is similar to non-competitive inhibition, but typically involves enzymes with more than one subunit. This type of regulation acts as an on-off switch for the activity of the enzyme. If an activator molecule binds to the allosteric site, it stabilizes the active site of the enzyme and allows the enzyme to function. On the other hand, if an allosteric inhibitor binds to the allosteric site, the active site is altered so the enzyme cannot function. Another way in which an enzyme can be activated is termed cooperativity, a type of allosteric regulation. When a substrate binds to the active site of one subunit, the active sites in the other subunits increase reaction rates. This is because the binding of one substrate causes a change in shape of the other subunits. The new shapes allow the active sites to increase catalysis. Feedback inhibition is a type of regulation also involving allosteric sites. Metabolic pathways frequently involve multiple enzymes, and feedback inhibition allows for the pathway to be shut off. A product formed by a metabolic pathway acts as an inhibitor. The product binds to the allosteric site of an enzyme at the beginning of the pathway, causing the pathway to shut off. Let's say the mall that holds the gumball machine is closing for the night. Since the customers are gone, the gumballs are no longer needed. The owner inserts the key into the lock, causing the enzyme to stop functioning. We have learned that enzymes increase the rate of reactions. They are essential for metabolic pathways, and their activity can be increased or decreased. Enzymes are constantly at work in your body, whether you are digesting a steak or sticking a quarter in a gumball machine.